Hello, my name is Brian Raux. I'm a medical science liaison at Biomerieux. In this chapter, I will be discussing how antimicrobial stewardship programs had some gaps opened up caused by the COVID-19 pandemic, and how we may be able to start to use diagnostics to bridge some of those gaps. Certainly, antimicrobial stewardship programs existed more than 10 years ago, but about that time, we really started to see a meteoric rise. In 2013, the CDC released their first antibiotic resistance threats report. In 2014, the CDC then called on hospitals nationwide to implement antimicrobial stewardship programs via these 7 core elements. They were leadership commitment, accountability, pharmacy expertise, action, tracking, reporting, and education. And at that time, about 41% of hospitals nationwide met all of those criteria. In 2017, the Joint Commission added their elements of performance for hospitals to meet with regarding antimicrobial stewardship. 2019 saw an update to that AR threats report. And then finally in 2020, the most recent year we have data for, 91% of hospitals are meeting all 7 of the core elements. I don't think I need to necessarily highlight for this audience the horrors of the COVID-19 pandemic, but these data, which are from about mid-August, show that there are over 590 million confirmed cases worldwide, which accounts for over 6.4 million deaths that were reported to the WHO. For many across the world, the demands that this put on the healthcare system were unprecedented. Stewardship programs, though, were well suited for this challenge. There were already existing relationships with infection prevention and control programs, so that stewardship programs could help with early case identification or assist with communication of positive results. Diagnostic stewardship, something that we'll talk about both here and in later chapters, is a critical component of any antimicrobial stewardship program as well. Because of the strong ties between antimicrobial stewardship and the microbiology lab, they're able to work together to work on the real-time interpretation of the various assays that have been developed over the last few years. Those test results can also be incorporated into the new treatment guidelines that many stewardship programs created and then redesigned again and again. The anticipation of drug shortages, which were both new and rapidly changing throughout the last several years, is something that a program needs to do, particularly when you keep in mind that many of the new therapeutic agents that became available were only accessible through an EUA, or emergency use authorization, and maybe each individual patient would need paperwork submitted to the institutional review board. The stewardship program could serve as a focal point for providers across the institution to submit those. Lastly, using their established skills, using prospective audit and feedback, stewardship teams could monitor and then enhance their treatment guidelines that they had started to put out. Unfortunately, though, while all of these things are great, resources are finite. Stewardship programs share their experiences on the implementation of some of those things as well as others. We know that New York City was a unique and challenging environment during the first wave of the COVID-19 pandemic. One stewardship program published their experience in 2021. This quote comes from New York Presbyterian, and they said that the early response to the COVID-19 pandemic did not change our antimicrobial stewardship principles, but instead changed our antimicrobial stewardship team's practices. I think diagnostics and antimicrobial stewardship go hand in hand. Each institution likely saw different gaps, and even the ones that shared the same gaps saw them to variable degrees. If we continue to advance and optimize the diagnostics that we have, we can use those to hopefully decrease the gaps that are brought about by antibiotic misuse. I'd like to move into the first gap that I will talk about in this series. This first chapter is going to focus on antibiotic misuse. So we know that throughout the COVID-19 pandemic, antibiotic use has varied across the different healthcare settings. Patients with COVID-19 frequently were prescribed antimicrobials despite lacking any efficacy for this viral infection. In the acute setting, in hospitalized patients, over the first 8 months of the pandemic, almost 80% of patients received an antibiotic. About a year later, despite overall antibiotic utilization being down, drugs that were used for conditions like community-acquired pneumonia, antibiotics like ceftriaxone, were still up. In the ambulatory setting, overall use in 2020 was lower than it had been in 2019. This is presumed likely to be due to decreased number of visits in that outpatient setting. But when you look about a year after the first set of waves, what you see is that use is back up to pre-pandemic levels, particularly again for those agents used for community-acquired pneumonia. While nursing home and skilled nursing facilities represent an overall small use of antibiotics, if you've taken care of patients from any of those settings, you know they can be a hotbed of resistance. So while antibiotic use rose and fell in these facilities with corresponding volume of patients, overall use was still down throughout the pandemic. In 2021, we saw those numbers come closer to what we had expected pre-pandemic. Stepping away from COVID for a moment to a broader discussion on the impact of rapid diagnostics on patient outcomes and thereby the antibiotics that they are receiving, I'd like to talk about a meta-analysis from 2017. This looked at the effect of molecular rapid diagnostic tests on the clinical outcomes of patients with bloodstream infections. It included 31 studies and had over 5,900 patients. And generally, they show that the clinical outcomes of patients with bloodstream infections favored those that had had a molecular rapid diagnostic test. The authors did a subgroup analysis where they looked at 26 studies that had the outcome of mortality included. And what they found is that the number needed to test with one of these assays to see a difference in mortality was 20. Really interestingly, they stratified these studies based on whether or not they had an antimicrobial stewardship program in place. And what they found was that when a stewardship program was in place, there was still a difference. When there was no program, however, shown in this second box here, that difference was no longer statistically significantly different. Data that include COVID-19 patients are still emerging. That could be another chapter in this series itself. As we get more data that are published, particularly on the impact of different diagnostics on patients with COVID, we'll see this literature emerge. But for now, this is one of the first studies that has become available. It's actually a performance study that was done after the implementation of one of these molecular rapid diagnostic tests. It was done in Italy in ICU or intensive care unit, patients that had COVID-19. They assessed 212 samples from 150 different patients that were suspected of having bacterial pneumonia. They had a positive percent agreement of 100% and a negative percent agreement of 99.2%. This reflects two different patients that had off-panel detections. What I really want to talk about, though, is the subgroup analysis that the authors did, where they actually compared patients that had both a positive molecular rapid diagnostic test and conventional culture, which is representative of the standard of care, which was 97 patients over 122 episodes. They compared this with the standard of care arm only, so this is conventional culture. This was in the first wave of COVID and before the assay had been implemented. So this was 29 patients with 31 episodes. Now, this is a subgroup analysis. The groups did have some notable differences between them, which are highlighted now here on the screen. SOFA score, how long they were in the ICU, how long they were mechanically ventilated. But the main outcome that the authors looked at was effective antibiotics within 6 hours from sampling. Patients that had the molecular rapid diagnostic test were more frequently on an effective antibiotic within 6 hours. In other studies, which was shown really by that meta-analysis on the previous slide, This has had an impact on outcomes. In this small study, though, that didn't flesh out. Neither in 14 nor 18-day mortality was there a statistically significant difference between the two groups. Again, it's a small population, a little bit unique populations, different waves of COVID. There are a lot of confounders here. So thank you. I'd like to wrap up this first chapter. We talked some about how antibiotic misuse may be affected by rapid diagnostic tests, both in this current pandemic, as well as how we can use that in the future. Next time, I want to talk about how this antibiotic misuse has started to impact antimicrobial resistance.